Good morning, honorary jury members, mentors, and fellow participants. I am Dr. Zubaida. My team ID is TM924UE9 and team name is Emma Insight. I am the team leader of my team, Emma Insight, and my mentee Aisha Kaiser has joined my team. The topic of my prototype presentation, entitled on From Molecules to Cancer Immunogenomics: Can Integrated Systems Biology Enable the Future of Precision Cancer Therapy? My vision emphasized to combine data science, systems biology, bioinformatics, and oncology and immunogenomics for development of innovative therapeutic and diagnostic strategies for gallbladder and gastric cancers towards an approach for one-size-fits-all by molecularly guided therapy. Despite these advances, several major challenges remain, like tumor heterogeneity results in diverse molecular profiles among patients, limiting the effectiveness of conventional therapies. Our software prototype, Nona ProBio, that integrates multi-omics data to identify cancer biomarkers and prioritize therapeutic targets for gallbladder and gastric cancer. It combines advanced softwares, including NCBI, UniProt, and the Protein Data Bank, PDB, for target identification. The nanobody design module employs AlphaFold, CollabFold, Rosetta, and PYMOL for protein structure prediction and optimization, while the molecular docking module uses AutoDock Vina, Haddock, and ClusPro to evaluate nanobody target interactions. Our project has 7 major objectives. First, we perform multi-omics analysis using in vitro NOZ gallbladder cancer cell lines and ex vivo human blood samples, and polyherbal plant groups for proper alignment codon sequence confirmation. Second, we design a polyherbal chimera using bioactive compounds from 12 medicinal plants. Third, we develop diagnostic chimeric nanobody from chimeric tumor antigens and fuse this nanobody with polyherbal sequence protein and also with GFP for cancer diagnostics. Fourth, we engineer Saccharomyces boulardii probiotic biofactories to express these nanobody polyherbal sequence protein therapeutic molecules and synthesize walnut polyherbal protein carrier matrix. Fifth, we integrate signaling pathway analysis. Sixth, we computationally study and predict apoptosis, DNA repair, oxidative stress, and extracellular matrix responses. Finally, we establish scalable recombinant production and delivery systems for future clinical translation. We target the gut-liver tumor axis and developing probiotic-based nanobody fused with polyherbal peptides to improve bile acid metabolism, detoxification, while modulate oxidative stress and inflammation in the liver by promoting anti-inflammatory cytokines to suppress the gallbladder tumor microenvironment, tumor growth, and enhance immune surveillance. Here, the immunosuppressive tumor microenvironment in gallbladder cancer depicts cytotoxic T cells, NK cells, dendritic cells, and M1 macrophages mediate effective anti-tumor responses. Our engineered probiotic will interfere with these immunosuppressive signals, reprogram the tumor microenvironment, and restore anti-cancer immune surveillance. We analyzed mitochondrial metabolic pathways involved in cancer progression. Our engineered probiotic peptide therapeutic strategy aims to restore mitochondrial function, reverse cancer-associated metabolic reprogramming, and inhibit tumor growth in gallbladder cancer. The human microbiome helps in maintaining health and regulating cancer progression. The gut microbiome communicates with host mitochondria through microbial metabolites, influencing cellular metabolism, energy production, inflammation, and immune responses. Dysbiosis can disrupt mitochondrial function, leading to metabolic reprogramming and tumor progression. Our engineered probiotic polyherbal peptide therapy aims to restore a healthy gut microbiome, improve mitochondrial function, and suppress gallbladder cancer progression through the gut-liver tumor axis. Our software prototype, Nona ProBio, have been designed to address these challenges into a unified molecular oncology system biology. In this prototype, we computationally identify, align, and get codon sequences from Curcuma longa, Berberis aristata, Scutellaria bicalensis, Rubia cordifolia, Falloppia japonica, Borhavia diffusa, Terminalia arjuna, Crataegus monogona, Bergenia ligulata, Paeonia officinalis, Valeriana officinalis, and Prunus mume. One of the most innovative components is our GFP-tagged diagnostic nanobody chimera, designed to recognize important cancer biomarkers such as EGFR, HER2, MUC1, CEA, MET, and VEGFA. Simultaneously, The attached polyherbal region delivers anti-inflammatory, antioxidant, anti-proliferative, anti-angiogenic, anti-metastatic, and anti-NF-kappa-B activities, creating a multifunctional therapeutic construct. We engineer Saccharomyces boulardii, a clinically established probiotic yeast, as a safe microbial chassis for targeted therapeutic delivery. The design gene construct incorporates the polyherbal chimeric nanobody expression cassette together with regulatory elements for controlled recombinant protein production. To enhance biosafety, the construct may incorporate biocontainment and regulated expression systems. Additionally, pathogen-targeted antimicrobial or antivirulence peptides can be expressed to combat clinically relevant bacterial infections while preserving the probiotic function of S. boulardii. The engineered probiotic functions as a living biofactory capable of targeted therapeutic delivery while modulating the gut-liver tumor axis. Our walnut lipid-based matrix carrier, which contains phospholipids, omega-3, omega-6, and omega-9 fatty acids, tocopherols, vitamin E, and phytosterols that encapsulate our designed polyherbal molecules and chimeric peptides, protects the bioactive molecules from degradation, improves targeted delivery, and increases bioavailability for cancer therapy. Excessive reactive oxygen species, ROS, trigger lipid peroxidation, mitochondrial dysfunction, and activation of the PI3K/ACT-NF-kappa-B signaling pathway, causing tumor progression. Mitochondrial release of cytochrome C activates CASPASE9 and CASPASE3/7, initiating apoptosis, and our engineered probiotic polyherbal peptide aims to reduce oxidative stress, inhibit aberrant NF-kappa-B signaling, restore mitochondrial function, and promote apoptosis in gallbladder cancer cells. The motivation behind our project is that cancer is not driven by a single mutation or pathway. Instead, multiple interconnected signaling pathways operate simultaneously. Therefore, rather than targeting one molecule, our systems biology approach integrates the PI3-KAKTMTOR, MAPKE-ARC, WNT-beta-catenin, NF-kappa, BTGF-beta P53 AMPK, and NRF2 pathways to achieve comprehensive tumor suppression while reducing drug resistance and improving patient stratification. Here are systems biology softwares for alignment for development of probiotic and nanobody polyherbal peptides after identification of cancer biomarker to reduce free radical-induced DNA damage and oxidative stress. We highlighted medicinal plants rich in bioactive phytochemicals with strong antioxidant properties that scavenge free radicals, reduce oxidative stress, and protect cells from DNA damage. These plants show promising therapeutic potential for the prevention and treatment of various cancers and form the basis of our polyherbal formulation. Rauwolfia serpentina provides bioactive indole alkaloids with antioxidant and immunomodulatory activities and inhibit angiogenesis, suppress cancer-associated fibroblasts, and reduce immune suppression within the tumor microenvironment, and activates dendritic cells, NK cells, M1 macrophages, and CD8 T cells, restoring anti-tumor immunity. Have included latest research paper and preprints. My published work on membrane-initiated PI3K-AKT nitrogen monofluoride Kb signaling pathway of programmed cell death also presented data of antioxidant enzyme and free radical formation demonstrating radiomodulation by Hocht 33258 combating hypoxia. Our prototype theme interconnects three research projects: the system biology-based polyherbal therapeutics, AI-enabled mitochondrial biology, and cancer immunogenomics, and integrates multi-omics analysis, mitochondrial metabolism, immune profiling, and bioinformatics to design chimeric antigen epitopes, polyherbal peptides, nanobody GFP, and walnut carrier matrix to support diagnoses and targeted treatment of gallbladder and gastric cancer. I will use this technology stack to develop my software prototype, NonaPro Bio. The front end is built with React, JS, HTML, and CSS, while the backend uses Python, FastAPI, and REST APIs for secure data processing and integration. PyTorch and scikit-learn support biomarker discovery, multi-omics integration, and nanobody design, with Postgres and AWS providing scalable cloud computing and data security. The software can be commercialized under the MIT license and Creative Commons 4.0, and deployed for hospitals, biotechnology companies, startups, research organizations, and academic institutions. Nona ProBio can also be offered as a software as a service, SaaS platform for biomarker discovery. Our long-term vision is to establish a globally scalable precision oncology platform for cancer diagnosis, treatment, and translational healthcare, with significant scientific, clinical, and commercial impact. I used insights from these references to develop and validate my prototype by establishing a scientific framework for constructing an integrated drug discovery pipeline that supports the computational workflow of our platform. Thank you very much for your valuable time and attention. I would like to acknowledge Royal Society of Biology, British Society for Antimicrobial Chemotherapy, Startup India, Inflib.net, ResearchGate. I would be happy to answer your questions. Thanks once again.